Klaas Enno Stephan is with us today. He is a professor both at the University of Zurich and at ETH Zurich, a professor for neuroscience. Welcome, Klaas, to this talk. You are a medical doctor and you are a computer scientist. How does that go together?
Well, the reason is very simple. For me, ever since I was a student, it was clear that if you do want to understand the brain and why it can break and exhibit a psychiatric disease, for example, then you do need mathematical models. There is no point in simply marveling at the complexity of the brain, either in terms of structure or its functional expression, and try to read meaning into the very complex patterns that emerge. You need a principled quantitative model that allows you to infer on the mechanisms that give rise to that complex behavior. So you need two things. You need to understand the biology, but you need to have the math and the computational skills to bring that into a model.
But why would the biology itself not be enough? If we understand how the brain is linked together and we know the synapses link up to these other synapses, why isn't that enough?
Because they're very, even for very simple networks, if you know every single connection, if you know every single node, it becomes impossible to predict how that network behaves very, very quickly. And the brain certainly has a complexity that goes far beyond what we can grasp with our minds. So computational models are totally necessary, indispensable for understanding how the brain works, particularly when you think about brain diseases.
Brain diseases such as?
Schizophrenia, depression, mania, bipolar disorder, obsessive compulsive disorder, these sort of things. Psychiatric diseases in particular, where you do not have a specific lesion, you can't, as opposed to many neurological diseases where you can see structural damage that the brain has experienced. In psychiatric diseases, typically there are very subtle or very distributed or no obvious structural changes at all. So there's something wrong with the functional organization. And to infer that, my belief is we need computational models. So for example, to give a counterexample, other people have promoted the use of genetics, and that, to my view, is a dead end. For the simple reason, we now know after decades of research that for any given psychiatric research, there are not only numerous genes that convey a predisposition and that interact, but critically, there is almost no disease where the genotype alone determines whether or not the disease will be expressed. But there is a very complicated and not understood interaction between the genotype and environmental influences, the things you've experienced in your life, say metabolic, immunological factors, but also life experiences associated with stress, for example. So we need something that lets us look at the endpoint of those interactions between the genome and the environment, and that means we must have models that we can use to quantify and understand how neural circuits at those endpoints of genomes and environmental influences, how they work in an abnormal way in diseases.
Now, how do I have to picture that? Are you're creating a simulation on some supercomputer where every single neuron is somewhere in there, programmed in there, and then you let simulations run through there?
No, not at all. Okay. We do that on our laptops, and hopefully at some point on our mobile phones. No, we take a rather different approach. There is no point in trying to build a replica of the brain, because then you're simply marveling at the same complexity that the real thing exhibits. You haven't learned anything. The art of modeling is that you need a clever reduction of the problem such that the mechanisms become visible from very high-dimensional, very complicated data. So our models typically don't look at single neurons, they look at the behavior of large populations of neurons, and they look at, for example, activity in one of these populations determines activity elsewhere via, of course, synaptic connections. Now the problem is, of course, in the human brain, we can't measure that activity directly. We can't stick electrodes into our brains and record from neurons as we can do it, say, in animals or in cell cultures. All we can use as clinically oriented scientists are non-invasive measures. For example, EEG, electroencephalography, where you record currents from the scalp, or fMRI, functional magnetic resonance imaging, where you put people in a scanner and you measure a signal that is related to the oxygenation of the blood, which in turn reflects the metabolic demands of neuronal populations.
Whether they are active or inactive.
Exactly, that's exactly that. So these data are rather coarse. They are not single neurons, they're not even small groups of neurons. They reflect activity changes in millions of neurons. And the trick now is you have to build a model that takes these data and computes or predicts what is the most likely underlying neuronal microstate, the underlying neuronal process that is most likely to have given rise to those data. It's a little bit like, say you're searching for oil. Yeah, you're out there in Alaska or wherever, you're searching for oil. You can't simply drill a hole 10,000 feet underneath, down. What you can do is you can just take some probes from the surface, say with ultrasound, and then again you need a mathematical model to infer what is the likelihood of oil being some thousands of meters beneath the soil. We do something very similar, but now with the brain. We record the activity in a non-invasive way from millions of neurons expressed in EEG fMRI, and we try to compute the most likely neuronal process that underlies these measures. And if we can do that, then we have in principle a handle on detecting disease mechanisms in the individual.
And how do you go about it? You have thousands of patients lined up, they will come to your lab, you know roughly what they have, and then you record all that through fMRI and EEG, or how does it work?
Yeah, not yet, we're not quite there yet. We've spent the past 5 to 10 years creating the methods for that computational modeling approach, and we have validated those tools and still are in the process of validating them using electrophysiological studies, pharmacological studies and patient studies. We are now in a position where we will be starting those patient studies, and we will have a dedicated unit for that here in Zurich, where clinicians and modelers will work together hand in hand. That'll be the first of its kind worldwide. But in practice, what we have done so far is we have tried to show that this approach works. And I'll just give you 1 or 2 examples. So for example, here's a silly example. An example, it's silly because your grandmother could solve the problem. Yeah. So what we've done is we have taken patients, and that's a collaboration with colleagues in London, a team led by Alex Leff. We have taken fMRI measurements of patients who have had a stroke. And as a consequence, they're suffering from speech disturbances, so-called aphasia. So there is no diagnostic problem. You just listen to these people and you know immediately who's had a stroke and who hasn't. However, because we now know precisely who is a patient and who is not when we take a control group, we can challenge our model with a rather tricky question. We can say, okay, here is data from the healthy part of the brain. Here's a measurement from the auditory system that is not affected by the stroke. We build a model that explains those data in terms of neuronal populations and how they talk to each other via synapses. And we fit it to that data from the healthy part of the brain. And now we ask, is the model capable of deciding, subject by subject, whether or not there is a lesion elsewhere in the brain, a remote disturbance that is reflected as a downstream consequence in the healthy part of the brain? And the answer is we can do it almost perfectly.
Wow.
With basically almost 100% precision. And you can now contrast that to any statistical approach that exists, and they are far, far worse. They're not much above chance. And this is just a very simple, and as I said, slightly silly in a way, illustration of the power of these models. Silly because this problem is a non-existent one, but here we have a ground truth situation. We can show that it works. Now you can imagine taking that approach and applying it to other situations where you are looking for, say, a very subtle and perhaps distributed lesion in the brain, and you want to know in a given patient, does this lesion exist, and of what sort is it? Because it doesn't have to be a structural one, it could be a chemical one. It could be a disturbance, say, in transmitter balances. And again, we have shown in studies that our models can detect this. This typically requires either animal studies, because you have to be invasive, you have to change something in the brain in a way that is not necessarily healthy, or in other cases we can use drugs in healthy volunteers and then ask the simple question, does the model detect which drug has been given, or whether drug or placebo has been given, or in some cases we know precisely, again from previous animal studies, what this drug does in the brain at a very microscopic level, say at the level of ion channels. So there was a study recently that was conducted in collaboration with my colleague Rosalind Moran, again at London, where she applied a drug called L-DOPA. That's something that you take for Parkinson's disease. It's a dopaminergic agonist, so it binds to dopamine receptors. Now, years of very careful animal research has shown in the past that when you apply dopamine to a certain part of the brain during a task of working memory, where you have to keep things in mind, then there are some very well quantified changes in the states of specific ion channels. You're changing the excitability of certain cells in a very precise way. Now, we did that with human beings. We used electrical measurements from the scalp, just electrodes in the scalp, very coarse measures, just waves of activity from millions of neurons, and we can show that under this pharmacological manipulation, with its known consequences, we can infer the same changes at the level of ion channels that have been shown previously with single-cell recordings in animals. So now we have a tool that we could use potentially in patients. And the sort of analogy that I often use is hypertension. Imagine you were a doctor 100 years ago. Every patient that came to you that had elevated blood pressure, as you could just measure with your little device, you would label hypertensive. It was one disease, just one thing. Now, over the decades, we learned that the very same symptom, namely rise in blood pressure, can be caused by completely different mechanisms in organs.
Such as?
For example, if you have a kidney disease, many kidney diseases lead to secretion of hormones that lead to an increase of blood pressure, to an increase in blood pressure. Or you could have a heart disease. You could have thyroid, hyperactivity of the thyroid, many other things. But they all impinge on the same mechanism at the end of the day, which is a tightening of your blood vessels. However, once we have tests to infer on in the individual patient, whether it is the kidney or the thyroid or the heart or whatever, then we can start treating patients in a mechanistic way. In the past, we just gave drugs to everyone that would just release the tone of the blood vessels, would just widen them. But that's just symptom-oriented, not causal. Now we can, at least for a proportion of the patients, decide, is it the kidney, is it the thyroid, is it the heart, etc., and then in many cases treat causally.
And today in psychiatry we're still at that level of 100 years ago.
Exactly.
Where we just record symptoms and we don't really know the underlying processes.
Absolutely. And that's exactly where I hope our models will help. Because once, at the moment, as you say, in psychiatry we are in a rather primitive state in that when we deal with an individual patient, we're just collecting a list of symptoms and we compare that against a classification, the so-called DSM-IV classification, that just says, for example, a patient shall be called schizophrenic if he or she fulfills 2 out of the following 5 symptoms over a course of 1 month. Now that means, and these symptoms, for example, are delusions, hallucinations, formal thought disorder, that includes, for example, disorganized speech, disorganized behavior, and then a 5th category is the so-called negative symptoms, things such as lack of motivation, blunted affect, those sort of things. Now, you just look at that list of symptoms, you can see immediately that there can be patients that don't even share a single symptom, and yet they're all called schizophrenic. And even those with the same broad class of symptoms can differ wildly. There are so many different types of delusions. For example, it's a very different thing if you have persecutory delusions and believe that your neighbor is from the CIA and is out there to kill you, than, say, the belief that someone has fallen in love with you. That is a very different thing, and perhaps very different mechanisms. So here, what happens is we just look at our patients, we record their symptoms, we group them into categories that have no validity whatsoever. And every psychiatrist knows they have no validity, but we have nothing else. And then we just treat them trial and error. We supply a drug or a psychological treatment or both, and we see whether it works. We wait a few weeks. If it works, great. Most cases it doesn't, and then we proceed to the next drug or psychotherapy, and we see whether that works. So what you end up with is that any given patient goes through often a rather long period until something is found that works, which means suffering for the patient, it's expensive, and it's very frustrating for everyone. So what we really want is something similar to the case as in hypertension. We want a test, we want a procedure that is objective, that we can apply to the individual, and that tells us something in mechanistic terms about the causes of the disease in such a way that not only we have a physiologically interpretable classification of the disease, but equally and perhaps more importantly, we can make a treatment recommendation. We can say with a certain probability this sort of drug is likely to work on this patient.
Now you're about to revolutionize psychiatry.
I would like to see that happening, but we will see whether it works.
Let's assume it works. No, you're on the way. So you're on the way. You're probably a danger to the established psychiatry establishment. How do they react when you come with your proposals and your thoughts? Are they scared because it takes their livelihood away, or
no? I haven't seen those sort of responses. By and large, they're quite positive in the sense that, as I said, most clinicians are acutely aware that something needs to be improved in psychiatry, that the current state is not satisfactory. Now, of course, there are major disagreements about how you could improve it. And clearly there are many colleagues in the field, including scientists, who find it difficult to get their head around my approach. And that is simply because, for example, they have been following other ideas for a very long time, so they've invested into other ideas like genetics. And that's perhaps the more important problem, clinicians in particular, but also biologically trained neuroscientists, are not used to think in terms of computational models. For them, that's an alien method. It's something they don't understand very well. So it takes time to explain to them why this is useful, and it takes convincing examples, which we're working on.
And how did you get into that? How did you... you're a medical doctor. Did you always have the feeling that there must be something there that we could solve with computational models?
Well, for me, in my life, it was actually almost the other way around, in that I started as a mathematician. Okay. So I started training in mathematics, and then there was a major crisis in my life, a broken love that made me rethink my entire life concept, made me leave town and change profession. I had always wanted to understand the brain, even when I started mathematics. But I thought mathematics would be a good start. Now at that point I decided I would switch to medicine, but at the same time do computer science. So in other words, change from mathematics to computer science, because at the time I was under the delusion that all you needed in order to understand the brain was basically to put all the knowledge you had about a single neuron, of which we do know a lot, into equations, and then simulate millions and billions of neurons, and the truth would somehow emerge from that very complicated construct. Now I realized very quickly that that was silly, because with that large mathematical machinery you can do anything. You can describe pretty much the universe, I would claim, but you have no way of testing whether what you simulate has any ground truth or not. For that, you need models that you can really challenge by real-world data and observations. So you must fit your models to data and ask how well do they explain and predict things in the real world. And patients are a great example of that. Additionally, when I then trained in medicine, I realized during my psychiatry training how, well, we talked about it earlier,
how primitive
yes, how difficult it is, how helpless we are, that we have so little to offer to the individual patient. It is really the area of medicine that needs most progress, and where I think the true revolutions will happen. in the next decades. That basically determined my trajectory. Yeah.
And now going forward, looking forward about 20 years, 30 years, where will we be? Will we be able to have a patient coming in to some clinic, get an fMRI or do some other tests, and then determine exactly what is needed and have personalized drugs to deal with that?
I would love to hope that. I don't think that will quite happen. I mean, we have to be realistic. If we look at, again, high blood pressure, despite more than 100 years of research, we haven't fully solved the problem. We can now treat many patients with high blood pressure well, and we can detect the causes, but there's still a rather large group where we still have no idea what's going on. And that is arguably a much simpler problem than understanding brain diseases. So personally, I would be happy if by the time that I retire, that some of our models have stood the test of reality and have managed to recategorize mental diseases. For example, take the very large population of depressed patients. It is a mass disease. If we can dissect out, say, 10 or 20% from those millions of people suffering from it across Europe, then that would be a tremendous and huge success. If we could not only understand physiologically in an individual what the problem is, but if we could also predict what the best treatment is. Same is true for schizophrenia, which has a lifetime prevalence of 1%. That is, each person has a 1% probability of becoming psychotic at some point in his or her life. And all those other diseases. So if we could start to redefine those diseases and then have models that are truly robust and sensitive and have stood the test of time in clinical practice, I would be very happy to see having that achieved at the end of my career.
Great. I wish you all the best.
Thanks very much.
Thank you.